Hello, everyone. My name is Abdullah, and today I will be talking to you about sequencing and localizing the nucleocapsid protein of tomato spotted wilt virus as the first step to understand the peanut beacon resistance in Alabama. So, as an intro about TWV, this virus is a single-stranded RNA virus. It belongs to an order called orthotusbovirus that also belongs to a family called Bunniaviridae. This virus has a global distribution, and that's because it's able to infect more than 1,000 plant species. The virus is being transmitted by at least 9 species of thrips in a persistent propagative manner. In 1988, this virus was first discovered in Alabama. After that, the peanut breeders bred for resistant lines, and several lines were introduced to us, and those lines been using for a long time. But recently, in 2021 and 2022, we noticed that the symptoms of the virus are increasing in the field. And that's why we basically wanted to do this study. So we wanted to understand why the peanut is observing high symptoms for the virus and what's happening exactly. We hypothesized that maybe there are mutations in the virus genome, which are pushing the emergence of new strains that are able to break the resistance of those lines. And to do that, we went to 3 locations in Alabama, BRU, as you see here, BRU, WG-RIC, and GC-RIC. These 3 locations are found here on the map, and also they represent the most counties that plant and produce a lot of peanut. We collected 126 samples from those 3 locations, and all the samples that were collected were showing symptoms for the virus of yellowing and also ring spot and also wilting. And to start this study, we basically wanted to start with the nucleocapsid, which is this very tiny gene on S segment. And the reason we selected the nucleocapsid is because it is used for detection for the virus in the field. And that's why it has a lot of data available on the NCBI. So all the samples were brought to the lab, and then the nucleocapsid was amplified from all of them. And then only the positive ones were sent for sequences. After that, we received the sequence and we made alignment. When we made the alignment, we used MT2 strain as a reference because it's a well-studied strain. After that, at the end, we selected 8 nucleocapsid to localize them in the plant cell, which will help us understand how the virus is functioning better. So as a result, we found 94 samples were positive out of the total collected, which was 126. This table here shows us the cultivar that were sampled from each location. All the cultivars that are listed in red, they come from the peanut RX, as some of them are listed there as resistant, some of them are listed as susceptible or tolerant. So what is the peanut RX? The peanut RX is the first guide or the first reference for the farmers before they select a certain cultivar to plant in their fields. And also peanut RX is the official institute in the United States that are responsible for breeding peanut resistant lines against multiple diseases, including TSWV. So these numbers to the right of each cultivar here, it refers to how many samples were tested positive out of how many collected. For example, here we found in TEF297, we found 3 positive out of 4 collected. And based on these results, most of the lines are very susceptible to the virus. The cultivars that are in black, they're still under development, are not released lines yet. When we made the alignment with MT2 strain, as you can see here, here's a map of the nucleocapsid that represent MT2, and here is a map of the nucleocapsid that represent all the nucleocapsid of Alabama. And we found 5 conserved amino acid mutations in the nucleocapsid. As you can see here, for example, in this site here, 21, the amino acid F in the MT2 strain, and changed it to L in our strains. And we call them conserved because all the nucleocapsid of Alabama, they show the same change. So here we have 5 conserved amino acid mutations. The phylogeny tree that we made, why we did it? Because it will help us understand our sequences, and it will show us the similarity or the relationship between our sequences and the rest of the sequences that are available in NCBI. So here are the 3 locations from Alabama, BRU, WG-RIC, GC-RIC, this big clade here. And as you can see, they're very close to Georgia, more than North Carolina or Asia. But most interestingly, we were able to see 2 subclades, A and B. These 2 subclades were mixed of sequences from the 3 locations of Alabama, and sometimes was just Georgia, and here was Georgia, North Carolina, and Asia. And in order to understand these 2 subclades, we selected 8 nucleocapsid, as you see here, they are marked with this red star, to localize them in a plant cell. So what is basically the localization? Basically, the localization is The localization is you have the gene of interest or the protein of interest, which is the nucleocapsid in this study, and then you tag this protein with GFB or a fluorescent protein. And then for this study, we use GFB. After you tag it with GFB, you infiltrate that in a plant cell, and then we image the virus under the microscope, and we see where it is being localized in the plant cell. This helps us to understand first the impact of the mutations on the localization and also how the virus is functioning. It's very important for us to understand how the virus is working in the plant so we can manage it. So when we did the localization for the 8-nucleocapsid, we found three main phenotypes. The first phenotype over the time course that was used, 2, 3, 4, and 5 days post-infiltration, we found in category number 1, the protein never aggregate. What I mean by aggregation is the protein movement in a cell. Does it aggregate? Does it move in the cell? Or is it very soluble? Actually, this one was very soluble and never aggregate. As you can see, until 5 days, it never aggregates. Sometimes it's also localized around the nucleus of the plant cell. In the second category, it was very soluble at 2 days post-infiltration, but then it started to aggregate, as you can see. You see a complete aggregation at 5 days. In the third category, it was very soluble at 2 days, but then it was aggregating faster than this category. So we saw a complete aggregation at 3 days post-infiltration. We used the free GFB as a localization control. So how these results help us to understand the main goal of this study is like we hypothesize that maybe we have mutations in the sequence that may given the virus its ability to break the resistance. So that was our hypothesis here, if you remember, and that's what we found. So when we did the alignment, we found 5 conserved amino acid mutations. We think that those mutations are one of the reasons the virus is able to break the resistance of those lines. Because when the virus is mutating, it's usually mutating for its benefit. So the virus can overcome the resistance of the plant and break it, and also keep replicating faster and be transmitted faster by thrips. So we think that those mutations are one of the reasons the virus is becoming more severe in the field. So when we selected those nucleocapsid and we localized them in the plant cell, we saw 3 categories. The first category, as I mentioned, it never aggregate. And when we looked here into these categories and the yield loss or the production from each cultivar, we found that the one that never aggregate, we didn't know the cultivar because the sample was collected in 2021 and it was collected by random sampling, which means we don't know the cultivar. So therefore we can't attract the yield production for this cultivar. But we did something else. So this one here, it says no aggregation number 4. But actually, when we looked into all the nucleocapsid that were amplified from Alabama, which was 140 nucleocapsid, we found that this mutation is not repeated or represented in any other sample, which means that it's not being selected by the virus. Therefore, it is suggested that this mutation is not good for the virus, or it is not responsible for the increase in the symptoms. Also, the faster aggregation, the plant was still given a very high yield here, which means even if the virus is aggregating or moving faster in the plant cell, it doesn't help the virus. But the second category, which is slower aggregation, you see a very low yield here, which means when the virus was aggregating slower or normal, it was actually given very low yield, which means the virus was very successful in this category. So we think that slow aggregation might be beneficial for the virus, and that's why maybe the virus is keeping mutating to make this mutation, which was presented in other 3 samples here. So maybe this mutation is the responsible for the virus symptoms increasing and the incidence increasing in the field. So how this study helps? So these findings should help the peanut breeders to breed for resistant lines in the future based on these findings. So they will breed better resistant lines that will resist the virus more in the future, hopefully. And the ongoing research right now, what we are doing is we're going for full genome sequencing, not just one gene as we did, but we're going to go for the full genome, the rest of the genome, and we're going to see if it has mutations or not. And this work was funded by the Department of Entomology and Plant Pathology, and also the National Peanut Board. And the facilities that were used to make this study is Dr. Martin's lab. Thank you so much all for listening, and I hope you find it useful. Please, if you have any question, email us on this email, and we will be very happy and to respond to your question. Thank you so much.